Hi everyone, this is just the recording from the end of class on Wednesday. Of course, it deleted itself, so I'm redoing it. First A, we're looking at the phospholipid molecules. These are the molecules in gray. Each one is a phospholipid because they have two parts to them. The hydrophilic is on the outside and hydrophobic is on the inside. The phospholipid bilayer is when these phospholipid molecules come together and form this bilayer here. So this is the bilayer. It allows for the passage of hydrophobic, nonpolar molecules. They also have to be very small. Cholesterol molecules are the ones in yellow. These help stabilize the cell membrane, so they keep these phospholipids together and don't allow them to condense too tightly to become rigid. The protein channels and pumps are what we see here in purple. They allow for the passage of hydrophilic, polar molecules. The other embedded proteins, such as receptors and enzymes, we can see here. And this one probably is too, an enzyme. We also lastly have cytoskeleton filaments. These are the spaghetti-like yellow strings that we see here that provide structure and stabilize the cell membrane. The cell membrane is selectively permeable. This means it can control or decide what can pass in or out. Why is this important? Well, it acts like a security. Make sure that nothing harmful to the cell can pass through, like pathogens. Substances can cross the membrane via either active transport, so through channels, or passive, which would be through the bilayer. In lab, we looked at a membrane, but it wasn't our cell membrane. In lab, the membrane allowed for distinguishing by size what was allowed through it or not. Our membrane is a bit more specific. It also will control, because of the bilayer, it controls both hydrophilic and hydrophobic molecules, or nonpolar and polar. In this chart that we're filling out, we're basing it off of these three ways it can pass through, either the phospholipid bilayer, the protein channels, carriers, or pumps, or through endocytosis or exocytosis. Starting with oxygen, it's nonpolar, very tiny. Because of these two properties, it crosses by the phospholipid bilayer. Glucose is a polar molecule. It's very small, but because it's polar, it has to cross through a channel. Unlike in lab, where it could just pass through the membrane through the holes. Starch, remember, is built up of glucose subunits. It's much larger, still polar, but because of its size, it has to be taken into the cell through endocytosis. Water is polar and small. Because it is really small, occasionally you'll see some water going through just the bilayer. But most times it has to come through a special channel called an aquaporin. Estrogen is nonpolar and it's small. This means it can pass right through the cell membrane. Insulin is a protein. It's soluble and it's large. Now, although it's soluble, it's very large. So in order to go in or out of the membrane, it has to go through exocytosis. So usually insulin is meant for binding onto a receptor to allow glucose to go through. But in the case of a cell releasing insulin, it's going to leave via exocytosis. Collagen is also a protein. It's insoluble. It's large. It's going to, because of its size, be exocytosed. SARS-CoV-2, a neat one that is relevant today. It's a lipid envelope, this virus, and it's very large. But this specific pathogen, in order to get into the cell, it has to bind to a receptor to allow it to go in. So once it binds, it will be endocytosed into the cell. In a normal cell, the extracellular concentration of sodium is high and inside is low normally. With respect to potassium, it's low on the outside of the cell normally and high on the inside. With channels, it is a passive way of diffusing through the membrane. Because it's passive, it's going to be moving from a high concentration to a low concentration for either potassium or sodium. So for potassium, because it's higher inside the cell, it's going to diffuse out. For sodium, it's higher outside of the cell, so it's diffusing in. Over time, though, this is going to balance each other out, and eventually it would be at an equilibrium where both sodium and potassium concentrations are the same inside and outside the cell. In order to correct this, protein pumps can be used to actively pump out potassium and actively pump out sodium and actively pump in potassium. This would restore the high concentration of sodium outside the cell and high concentration of potassium inside the cell. Why can't ions pass through the phospholipid bilayer directly? Because ions are charged. These notes will be put up on Moodle for you to copy anything out.